Okay, now that we've learned about carbohydrate monomers and polymers, we're going to talk about how they can work in conjunction with proteins to have different functions biologically. And so we're going to talk about carbohydrate protein mixtures. Mixtures isn't really the right word. That suggests that these are separate from each other. But when protein and carbohydrates are bound together in the complex, we get three categories: peptidoglycans, glycoproteins, and proteoglycans. The way we can think about these is really the end of the word talks about sort of the parent molecule. So a peptidoglycan means that it's mostly sugar and it's got a peptide, a small amino acid sequence attached to it. Glycoproteins are mostly proteins, so they're proteins that have small sugars attached to them. And then proteoglycans are sort of a more equal combination of proteins and sugars. So let's start off by talking about proteoglycans, mostly sugar with small peptides on them. And those are the molecules that our lysozyme is recognizing that are part of the bacterial cell wall. This is one good example of a peptidoglycan. The sugar on the bacterial cell wall is this NAM-NAG polymer that we've been talking about as the substrate for lysozyme. And here are the monomers of NAM and NAG, right? Here's our NAM, right? And here's our NAG. And all they are is really glucoses that have been modified to have either amide groups on them or amide groups plus a carboxylic acid. It's not important that you know the structure of NAM-NAG polymer, but it's pretty much a modified glucose. And we can figure out that these are 1,4 linkages between the NAM and the NAG, and these are beta 1,4 linkages because the oxygen that's bridging is on the same side as the sixth carbon. Now, if we look at the peptidoglycan in the bacterial cell wall, this is a staph bacteria. We can see that we have these long polymers of NAM-NAG that make up the sugar, which is the main part of the peptidoglycan. But these long polymers of NAM-NAG, shown here in red and green, are sort of cross-linked to each other with short amino acid sequences. Right? And these bridging amino acid sequences are alanine, glutamic acid, lysine, alanine, and then there's a long stretch of glycines. And this helps bridge these long strands of NAM-NAG and sort of solidifies this bacterial cell wall as a solid surface. Now we know that lysozyme comes in and it recognizes the bridging oxygen and cleaves the NAM-NAG polymer, disrupting this organization and lysing the bacteria. There are also, there's a long metabolic pathway here shown that generates these peptides that link the NAM-NAG polymer in bacterial cell walls. There's lots of enzymes that sort of couple these amino acids together. Here you can see this peptide being built. This is the peptide that gets coupled to that stretch of glycines that bridges the NAM-NAG polymers. And some antibiotic agents actually target, as inhibitors, different enzymes in this pathway. And you can see why things like penicillin or bacitracin or bactoprenol, these inhibitors of the enzymes that are involved in the metabolic pathway that build this peptide in the peptidoglycan of the bacterial cell wall would be effective inhibitors of bacterial growth, right? Penicillin inhibits this particular step where alanine is shuttled off before you make your final peptide in here. So it's a good target, right? We don't have bacterial cell walls. We don't need these enzymes that are used to construct the peptidoglycan that's vital for the cell wall integrity. And so if we can develop drugs that are inhibitors of the formation of this peptide, then we can inhibit bacterial growth. Here's penicillin structure over here. It's an inhibitor of this enzyme. I don't even know what it's called over here, but enzyme number 9 in this big, long metabolic pathway. But of course, some bacteria get around. Um, they have antibiotic resistance, and that's a huge problem clinically now. And there's a set of enzymes that bacteria can produce called beta-lactamases that cuts this bond in penicillin, making it ineffective as an inhibitor. And so bacteria that are resistant to penicillin tend to generate these beta-lactamases to destroy that inhibitor. Okay, so that's an example of peptidoglycans, mostly sugar with some peptide cross-linking. Proteoglycans are mostly um, are an equal mixture of proteins and sugars, and a lot of times these proteoglycans are present on the outside of cells in the extracellular matrix, and they have some sort of structural role. So here's a sort of cartoon that shows all these proteoglycans, proteins that are bound on the outside of the plasma membrane that have these sugars sort of decorating them, that are pointing out into the extracellular matrix. So a lot of these proteoglycans don't have real specific functions, but they are really structural to maintain the integrity of the plasma membrane. Okay, the last one's glycoproteins. We just got proteins that we've stuck some carbohydrates on. Now, this you can think of as a post-translational modification of a protein. Protein is translated and then sugar is attached to it. And just like any post-translational modification, that can affect the function of the protein. Some proteins require glycosylation in order to be functional. Some are inhibited by glycosylation. And so the presence of this sugar can have a real effect on the function of the protein. So let's look at some examples. Oftentimes, glycoproteins are involved in the immune response, and that's because having all of these interesting monosaccharides with various linkages, alpha and beta, 1,6, 1,4, can create carbohydrates that are really unique and almost act as a fingerprint for cells to be identified or to bind to specific receptors. And so here you can see an immune response to inflammation and how T lymphocytes use glycoproteins in order to bind to sites of inflammation. They have a bunch of glycoproteins on the outside of the lymphocyte, and the model is that they sort of roll down the interior surface of a capillary, and if there's inflammation, there's receptors that are secreted or bound to the inside of the capillary that can potentially bind these glycoproteins. And so as this lymphocyte sort of rolls down the inside of the capillary, if there's enough receptors that bind to these glycoproteins, it'll stop and it will release some anti-inflammatory agents or react to this site of inflammation. And so the glycoprotein is important, and the glyco part of it is important because it provides this specific recognition that these receptors bind to. And carbohydrates are really good at that. Here's some other examples of glycoproteins being used in cell-cell recognition. It's really similar to that last example. We've got these proteins that are membrane-bound proteins in these cases that have these interesting branched polysaccharides, heteropolysaccharides. And these, again, can act as a real unique identifier for a particular cell and can bind very specifically to particular receptors. In this case, we've got a virus that recognizes a glycoprotein that's being displayed on the outside of the cell, and that probably allows this virus to infect this particular cell. Some toxins recognize glycoproteins, bacteria, and again, we've shown how lymphocytes are able to recognize cells. There's a lot of research going into stem cell identification and purification. And really, in order to purify stem cells, you look for a particular glycoprotein signature that's displayed on the outside of the cells that's specific for stem cells.